Hayley and my project is on the influence of relative humidity, temperature, and light cycling on winter tick questing at 4 geographic locations. So what is questing? Questing is the behavior demonstrated by a tick larvae when they crawl up vegetation in search of a host. Here on the left we show what questing looks like in nature, and here on the right is what it looks like under our experimental conditions. This is important because this is the time that the larvae will actually attach to a host, which they can ultimately at that time transmit diseases. So if we understand how the diseases are transmitted from ticks that attach to moose, which are the ones we studied, we can correlate this to how they may transmit human diseases as well. So this experiment will show what factors can influence the process of questing. So for this experiment, we wanted to see what factors actually trigger this behavior. We wanted to know if it was driven by the environment or by endogenous molecular signaling. So we set up these incubators here that I showed an example of on the right, and we want to look at 3 variables, specifically temperature, relative humidity, and the photoperiod, or how much light they're exposed to during a day. So this is a closer look at the study design. We used approximately 50 larvae from 5 different females from 4 sites, which is shown on the map here. We used Maine, Texas, and Idaho and Wyoming, which are relatively close together. We used 4 different incubators, 2 of which were kept at constant temperature at 22 degrees Celsius, and 2 that alternated between 2 and 20 degrees. And each of these temperature groups had 2 different light cycling periods, one that had 12 hours a day, 12 hours of dark, and then the other one that had 10 hours a day, 14 hours of dark. This here on the right is an example of the test tube rack that was put into each incubator. The top half was the 50% relative humidity group, and the bottom half is in 93% humidity. And then each of these 4 rows represents a site, and each of these 5 test tubes are the individual females. This is a closer look of how the vials themselves were actually set up. We used a larger test tube here with the salt solution in the bottom that generated either the 93% or the 50% relative humidity. And then this is the actual test tube that we put inside of this one. This bottom one here is where we initially put the tick larvae. The bottom is cut out and covered in mesh so that the salt solution can actually reach the ticks. And then over time they were allowed to crawl up this middle tube and into this top tube. Here on the right, we show what it looks like when the ticks are not questing versus when they are questing. So throughout the weeks of the experiment, we measured the proportion of the ticks questing that moved from the bottom vial to the top vial by weighing them. So while analyzing our data, we used a generalized additive model to look at the proportion of individuals questing as a function of the different sites and treatments. On the y-axis, we show percent questing, and on the x-axis is time. These 4 graphs on the left are the 2 to 20 oscillating temperature groups, and these 4 graphs on the right are the constant 22 degree temperature groups. We have our 50% relative humidity groups here shown by the circles, and the 93% in the triangles, and the same for the 22 degree group. And each of these colored lines represents one of the 4 sites. We can see here that there is a significant difference between the ticks that were held at the 22 degree temperature group, which did not quest at all regardless of the humidity or the site that they came from, and here on the left, the 2 to 20 degree groups definitely had a significant amount that quested. So now I want to take a closer look at the 2 to 20 degree temperature groups, because this is where we can really see the effects for other variables. These graphs on the right are the same axes and groupings as before, just zoomed in on the oscillating group. Here we can see that there is a significant effect of site. Even though there isn't much of a difference between Wyoming and Idaho, we would expect that because those sites are so close together. This tells us that endogenous molecular signaling must play a role in triggering questing. If it didn't, then all of the treatment groups, regardless of site, would have the same pattern on the graph. This also tells us that there is a significant difference between the relative humidity, because we do see a lot more questing in these 50% humidity groups rather than the 93% humidity groups. This also shows that there isn't a difference in the short day versus long day periods. We did not find that there was a significant effect of day length for triggering questing. But since there is differences between the site groups for each of these environmental conditions, This does tell us that there is a significance of environmental effects for triggering questing behaviors. So the results of this experiment show that both endogenous molecular signaling and environmental factors both play a role in triggering questing behaviors. We see the role of endogenous molecular signaling by the differences among sites, and then we see the effects of environmental factors by the differences we saw in relative humidity and increasing the temperature variability. This makes sense because fall is characterized by lower relative humidity and oscillating temperatures, and this is the time of year that may trigger ticks to quest and begin searching for a host. Ultimately, this experiment will help us narrow down conditions and hopefully create models that will tell us when ticks are most likely to be questing, so we can target control measures for when we know they will be at the top of vegetation, such as spraying insecticides or mowing. I want to say a big thank you to Dr. Benoit and Dr. Oyen for allowing me to be a part of this project, and for our other collaborators for making this project possible. Thank you.